First of all, there are two groups of white blood cells, the granulocytes and the agranulocytes. The granulocytes are basically the phils, the neutrophils, eosinophils, and basophils, and the agranulocytes are the lymphocytes and the macrophages. So you should know the total white blood cell count, which is 7,000 per microliter, which is the average, and also the percentage of the different white blood cells in the blood. The granulocytes and the monocytes are produced only in the bone marrow, while the lymphocytes and their plasma cells are produced in the different lymphoid tissues like the spleen, thymus, the tonsils, pars, patras, the small intestine. Once the granulocytes are released from the bone marrow, they stay about 4 to 8 hours in the blood and then another 4 to 5 days in the tissues. The monocytes stay only for 10 hours in the blood, and after that they enter the tissues where they swell to form macrophages, and the macrophages can live for many months in the tissues. Lymphocytes, basically, they enter the circulation, and then after that they can enter the lymph, and from the lymph they can go back into the circulation. So this cycle continues, and the lymphocytes can live for weeks or months, depending upon the body's need for these cells. The neutrophils and macrophages are important in the defense against infection, and they have common functions or features, you can say. Both of these cells show diapedesis, amoeboid motion, chemotaxis, and phagocytosis. The difference between them is that the neutrophils are considered mature cells because they can do phagocytosis even in the blood, while the macrophages are not mature cells because monocytes have little ability to phagocytose in the blood, but once they enter the tissues and they become macrophages, they have greater ability to phagocytose invading particles. So basically, diapedesis, amoeboid motion, and chemotaxis all occur during inflammation. Diapedesis is basically the squeezing of neutrophils and monocytes through pores in the blood capillaries, and amoeboid motion is basically the process by which the neutrophils and macrophages move through tissue spaces, and their velocity is 40 micrometers per minute. And chemotaxis is basically the process by which the neutrophils and macrophages are attracted to the site of inflammation by chemical substances produced by the inflamed tissue, and these substances can be bacterial or viral toxins, degenerative products from the inflamed tissue, products of the complement system, and reaction products caused by plasma clotting in the inflamed area. So the most important function of the neutrophils and macrophages is phagocytosis. The body's own cells and tissues are not phagocytosed because of their smooth surface and a protective protein coat which repels the phagocytes. So for a substance to undergo phagocytosis, it must have a rough surface and it must have no protective protein coat. Also note that the susceptibility of bacteria to phagocytosis is increased due to the involvement of complement factor C3, and this is known as opsonization. Basically, in opsonization, the bacteria or any other invading agent is made susceptible to phagocytosis by complement proteins. So now let's talk about the difference in phagocytosis between neutrophils and macrophages. As you know, the neutrophils are mature cells and they can begin phagocytosis immediately. However, the neutrophils are not as powerful as the macrophages because they can only phagocytose 3 to 20 bacteria. After that, they become inactivated and they die. While macrophages are much more powerful because they can phagocytose up to 100 bacteria, along with red blood cells and also malarial parasites. And even after phagocytosis, they can survive for many months. So the lysosomes in both neutrophils and macrophages have proteolytic enzymes for the digestion of the bacterial cell walls and other foreign proteins. But the thing to note is that in macrophages, the lysosomes also contain lipases, which are not found in neutrophils. So these lipases are important for digesting the thick lipid cell membranes of the bacteria, such as that of the tuberculosis bacteria. So even if the lysosomes and neutrophils and macrophages cannot digest the bacteria or other foreign particles, these cells have bactericidal agents which can kill the bacteria. And these agents are basically oxidizing agents like superoxide ions and hydroxyl ions and hydrogen peroxide. And these are found in peroxisomes and in the membrane of phagosomes. This is important to know because the tuberculosis bacillus is especially resistant to lysosomal digestion. The reticular endothelial system. It's basically a generalized system of phagocytes which is present in all tissues, and especially in those tissues where large amounts of foreign particles, toxins, and other substances must be destroyed. It includes all the monocytes, the mobile macrophages, the fixed tissue macrophages, and some specialized endothelial cells in the bone marrow, spleen, and lymph nodes. So the macrophages involved in this system include histiocytes present in the skin and subcutaneous tissues, the alveolar macrophages present in the lungs, the Kupffer cells present in the liver sinusoids, and other macrophages present in the lymph nodes, spleen, and bone marrow. So the thing to note here is that once macrophages enter the tissues, they can attach to the tissues and remain attached for months or years until there's some kind of infection or inflammatory response, and after that they break away from their sites of attachment to take part in that response. The alveolar macrophages have the ability to form a giant cell capsule around those particles that can't be digested, and so these particles remain trapped in this capsule until they can be slowly dissolved. And this capsule is usually formed around the tuberculosis bacilli, silica dust particles, and carbon particles. The macrophages of the spleen and bone marrow are involved when the invading organism happens to enter the blood. And when this blood passes through the spleen, the macrophages in the spleen will phagocytose the invading organism. And at the same time, the macrophages also phagocytose old and abnormal RBCs. Inflammation basically means the dramatic secondary changes taking place in surrounding uninjured tissues due to substances released by the injured tissue. So that if this is the injured tissue, the substances released by this tissue will cause effects in the surrounding tissue, resulting in inflammation. Tissues can be injured by bacteria, trauma, chemicals, or heat. Inflammation is characterized by vasodilation of local blood vessels, increase in permeability of capillaries, leakage of fluid from those capillaries into the interstitial spaces of the tissues. So this results in edema and clotting of fluid in the interstitial spaces because of leakage of fibrinogen from the capillaries, and also the migration of large amounts of granulocytes and monocytes into the inflamed tissue. The substances responsible for causing this inflammation are produced from the inflamed tissue, obviously, and these substances include histamine, serotonin, bradykinin, prostaglandins, products of the complement system, and reaction products of plasma of the blood clotting system, and also lymphokines, which are secreted by the activated T cells. So because of all these chemical substances, the effects of inflammation are produced. Now, the walling off effect of inflammation means that the area of inflammation must be walled off, or it must be blocked off from the surrounding tissues to prevent the spread of the inflammation. So this happens when the tissue spaces and the lymphatics in the inflamed area, they are blocked by fibrinogen clots, and so the flow of fluid in the tissue spaces and the lymphatics is slowed down, and this prevents the spread of the bacteria. The neutrophils and the macrophages are the major white blood cells involved in inflammation. The inflammatory response is divided into 4 lines. The first line of defense is provided by the tissue macrophages. which include the histiocytes, the alveolar macrophages, and the other kinds. These tissue macrophages are activated by the products of inflammation. They undergo rapid enlargement, and they begin phagocytosis within minutes. And the fixed tissue macrophages, they break away from their sites of attachment and they become mobile. The second line of defense is provided by the neutrophil invasion of the inflamed area, and this occurs within the first hour after the inflammation begins, and it is caused by the release of cytokines by the inflamed tissue, like interleukin-1 and tumor necrosis factor alpha. And the function of these cytokines is that they cause margination, diapedesis, and chemotaxis of the neutrophils. So margination basically means is that they cause the neutrophils to stick to the capillary wall, and diapedesis means squeezing through pores in the capillary wall, and then chemotaxis is basically the movement of the neutrophils towards the source of the inflammation. So here you can see margination, diapedesis, and then chemotaxis towards the inflamed tissue. And during the second line of defense, there is an acute increase in the number of neutrophils, which is called neutrophilia. So the number of neutrophil increases from the normal count of 4,000 to 5,000 to 15,000 and 25,000 per microliter. And this is basically caused by the products of inflammation. These products of inflammation act on the stored neutrophils in the bone marrow and mobilize them. They enter the blood and then they go towards the inflamed tissue. The third line of defense is provided by the second macrophage invasion, and this happens when monocytes from the blood, they enter the inflamed tissue and they swell to form macrophages. Now this process requires up to 8 hours or more, and the buildup of macrophages is slow, and it takes several days or weeks, and after that the macrophages become the main phagocytic cells. The fourth line of defense is provided by increased in production of granulocytes and monocytes by the bone marrow, and there is an increase in this production because activated macrophages produce certain factors, and these factors include granulocyte colony stimulating factor, monocyte colony stimulating factor, and GM colony stimulating factor, or you can say GM-CSF. So all these factors, they act on the progenitor cells in the bone marrow, the granulocytic and monocytic progenitor cells in the bone marrow, and eventually this leads to the production of increased granulocytes and monocytes. So here are these factors that we were just talking about, and these are produced by the activated macrophage. These include GM-CSF, G-CSF, and M-CSF, and as well as tumor necrosis factor and interleukin-1. And all of these act on the bone marrow to increase the number of granulocytes, to increase the production of granulocytes and monocytes.